Now let's take a look at summarizing some of things that happen with a mitochondrial dysfunction. What's going on in our mitochondria? We know that they are especially prone to DNA damage. And there are even whole theory, the mitochondrial free radical theory, that suggests that the more we consume and eat, the more cell respiration that goes on. Thus, the more oxygen is split in order to form water, which generates when we split the oxygen free radical that can go around. And to damage, the more the damage that's done, the more that aged cells become. Now, there have been some challenges to this free radicals theory, or expounding upon that free radical theory. It really depends on how you look at it. But this is how I will summarize that information. Turns out that not only do we have the idea that yes, indeed, mitochondria do accumulate free radical damage throughout their lifetime, and that becomes more with aging. But it's purely because of the increase in free radicals or oxidative reactive oxygen species. So the question is, do these species actually cause our deterioration, or what else might be going on? More recent events suggest that perhaps the mitochondria, in response to stress, right, if they are not functioning that well, or if there are not enough energy around, upregulate the production of mitochondria such there are many more mitochondria. And as a consequence of that, the more mitochondria are producing more reactive oxygen species, or one and a half of oxygens that are able to go around and do more damage, but also increasing the energy output. So might that anyway, as a congruent thing, we are growing more mitochondria in each cell. They are also producing more reactive oxygen species. So perhaps the increase in reactive oxygen species, or ROS, is due to the fact that there are simply more mitochondria. Either way, the reactive oxygen species are causing damage and a reactive aligned with cellular aging. So just a little note on some of the newer research, but free radical theory is sort of leading idea at the moment. Moving on to the next physiological trait of aging, you are probably very familiar with the idea of telomere attrition. Certainly, we have covered the telomere shorten as cells go through divisions. Stem cells and cancer cells might have telomere active, so they can continue to grow or regenerate telomeres. However, most somatic cells lose that capacity. And you are probably familiar with the Hayflick limits. Hayflick limits suggest that there are limited number of cell divisions that somatic cells can go through before the telomere becomes short enough to start doing damage to the telomeric region of chromosome 1 of telomeres. Essentially run out, it's suggested that 50 division is about the maximum for any somatic cell. So after that, each cell division is shortening the ends of the chromosomes until we start actually nibbling away at the genes located on the ends of chromosomes, which, as you can imagine, can have some manifestation in the condition of the cell. So telomeric attrition definitely associated with the aging cell.